Okay, so today I'm going to tell you a little bit about a project called ENIGMA. It's a very large-scale international study of the human brain. And I think the talk will last maybe 50 minutes or so. There'll certainly be some time for questions at the end. And also, I mean, if you stay at USC, you're very welcome to work with us on this project. It's one that's always open for new folks who are skilled in neuroimaging. So just to start off, here we go. So a lot of us are very interested in something called population-based brain mapping. And so we're interested in looking at a large number of people around the world and understanding, how does the brain change throughout life? Can we map the effects of disease or treatments or certain risk genes on the way that our brain structure and function change throughout life? And as all of you know, you can look at the brain with a number of different imaging techniques. So there'll be modalities that look at the anatomy, the structure of the brain, the molecular composition, the activation and the activity. And really until quite recently, we didn't know a lot about the relationship between, for example, genetic variations in our DNA and the types of changes that were going on in the brain. So that led to a field known as imaging genetics. So there's also been a huge rise in the development of biobanks. And so many of you will be familiar with the UK Biobank and other types of imaging repositories. So basically around 10 years ago, ENIGMA started to publish the largest neuroimaging studies of 9 different brain disorders. And the question we all wanted to address was, what are the factors that are helpful or harmful to the brain? This has grown to analyze brain scans and genetic data from a little over 100,000 people now from 45 countries, over 500 medical centers. And right now there's about 2,000 scientists organized into over 50 different working groups that are really studying different questions about the brain. So I'll tell you a little bit about the largest neuroimaging studies to date of some of the major psychiatric and neurological conditions that we are interested in. You'll see bipolar disorder, major depression, schizophrenia, but also obsessive compulsive disorder, ADHD, autism spectrum disorder, PTSD, epilepsy, and some neurogenetic conditions such as 22q deletion syndrome. And I'll tell you a little bit about how they're set up as international projects. So one of the reasons we wanted to develop a sort of big data international project is that it allows you to answer new kinds of questions. And so one of them might be cracking the brain's genetic code. So what are the areas or markers in the genome that influence how a brain is wired, how it changes through life, through the lifespan into old age. And these were just impossible to solve before. There just was not enough information that any single person had collected. So that could be called a sort of power or statistical power advantage. The other reason is efficiency. So I mean, if you could compute measures from brain scans at 500 different sites around the world in 45 countries, you could do a lot of cooperative work. You could do machine learning in interesting ways, deep learning in interesting ways. You could apply new mathematics. I mean, anyone can think up a new algorithm that they have developed to query these data. And then the final advantage is team science. So a lot of these papers might have 300 or more authors that are in Nature Genetics, a lot of these top journals. And that's something that we often would call collective intelligence. So, I mean, no one of us knows all the answers on our own, but by working with a lot of other people with different backgrounds and skills, I mean, some of these questions become a little bit easier to solve. So there were 2 main goals when ENIGMA was started 10 years ago. So one of them was, how do the major disorders of the brain affect the brain? And I mean, you could look at them with MRI or diffusion tensor imaging or resting state functional MRI or EEG. And we just want to know, I mean, what is the difference between a person with a particular disorder such as Alzheimer's or Parkinson's and everyone else? And then a little bit trickier of a question is, how does variation in our genetic code affect the brain? So we screen millions of markers in our genome. There's different techniques, genome-wide association studies, epigenome-wide association studies. CNVs are copy number variants, and you can look at these different types of variations in the genetic code and see what difference they make to the brain scans by compiling vast amounts of data from around the world. And one reason we'd like to do this is we want to understand genetic markers and maybe other factors in the environment and elsewhere that affect our brain. So our development and aging and risk for disease and how we respond to a treatment, they might depend to some degree on our genetic code. So we want to know about that. So here's a map of the ENIGMA members. The colors are the disease that each group studies mainly. So if you see a little blue bubble there for ADHD, the site is scanning people with ADHD or autism or addiction or a number of other things. And I think, as you can appreciate, obviously a lot of these sites have been scanning patients for many, many years. And collectively, I mean, you could really analyze data sets of tens of thousands of people. You just have to sort of coordinate the analysis in a way that everyone can participate in easily. So to organize the work, there are 51 working groups. They're run by working group chairs, there are 95 of those. There's 33 different diseases and disorders that are studied. And there's actually a little key to the disorders at the bottom that are studied there. And obviously there's a great deal of data that's being collected from imaging on those across the world. So if you want a quick review of what ENIGMA has done, you can actually take a picture of that QR code with your cell phone. And if you do, it'll take you to a link to a review that was written by people all over the world talking about what they've achieved through doing this. There's also a couple of overview articles, one in Science from a couple of years ago and one in Neuron from a couple of years ago, just talking about what the goals and benefits of the international collaboration are. So if you're in ENIGMA, you might be interested in a particular disorder. This is a sort of roadmap of the different disorders that ENIGMA is studying with brain imaging. So up at the top left, there's major depression, bipolar, and schizophrenia, all major psychiatric conditions. Related to schizophrenia, there are other disorders, early onset psychosis, schizotypy, and people who are at very high clinical risk of schizophrenia. And you can imagine that you could study any of these with imaging at multiple sites. There's dedicated working groups on anxiety, panic disorder, generalized anxiety, PTSD. In the neurology field, there's dedicated working groups on brain injury, ataxia, epilepsy. And initially, all of these groups are trying to figure out what it is about the brain that's different in their disease of interest. And then later on, they might be able to compare each of the disorders they study with some of the others. Now, this is a little bit of a simplification. So if you look in these boxes, even the group of brain injury within ENIGMA has many, many subgroups within it. And you can imagine why. So if you have a brain injury, you might be in the military, or you might be concussed as an athlete playing sports, or if you have children, your child, I hope not, but might have a brain injury. And you can imagine that each of the ways that you might get a brain injury might differ in how the outcomes play out or how they look on a brain scan. So all of these groups are sort of organized into different subgroups that tackle different types of aspects of the conditions. And then there are also some non-clinical groups. So many of you know about MRI, diffusion imaging, resting state. If you're going to analyze data from all sorts of different scanners, there's a lot of algorithms that are used routinely, things like FreeSurfer or FSL. But there's novel things that you can look at. I mean, if you're looking at genetic data or epigenetic data, there might be algorithms for looking at those things like epigenetic age. So if you look at this picture, there's technical groups that develop code and algorithms that the other groups use to try and derive information in a harmonized way from the scans. So here's some of the people that lead the ENIGMA groups. You'll probably recognize Neda and some of the other people that are shown there. But to be honest, the majority of the work is done every day by people all over the world, people from different countries you'll see here, Thailand, Russia, Australia, Korea, I mean, even countries that don't traditionally work together, such as Russia and Ukraine, and many others are working together on this. And just to sort of talk about how they work together, and if you had, as you see in this slide, 4 scientists that are working on a certain disease, they might cooperate with another group of scientists that are working on other diseases. And then, if you look at this, there might be scientists analyzing anatomical MRI or DTI or resting state functional MRI. And if you look at this sort of network of the red dots are the scientists and the links are sort of collaborations or information, you can imagine how a group that's studying diffusion imaging of Parkinson's disease might be able to help out another group that's studying diffusion imaging of Alzheimer's or frontotemporal dementia. So this sort of scientific cooperation is known as a modular hierarchical network. I mean, there's a lot of bridges and links between different projects, and that's really helpful in growing a large international study. So one of the first achievements of ENIGMA was just to map how do the different diseases that I mentioned affect the brain. And I mean, first of all, just with structural MRI, all of us know that the gray matter of the brain changes throughout life. A lot of these conditions, such as major depression, bipolar disorder, schizophrenia, patients tend to have an abnormality in the cortical gray matter. And these are just pictures of the level of gray matter loss on average in people with these disorders. You'll see OCD, ADHD, autism spectrum disorder, and some really interesting things. I mean, most of the disorders are associated with a reduction in gray matter, but not all of them. If you look at autism, ASD, they're in the middle of the right column. People with autism may tend to have an excess of tissue in the frontal lobe. So the blue colors show a sort of excess or larger amount of gray matter than is typical in match controls. So I think you can begin to sort of appreciate the diversity of these illnesses just by looking at these sort of overall pictures. And you can buy a T-shirt. There's a T-shirt with all these ENIGMA diseases on that Jan Boitelaar, who runs he co-chairs the autism working group, he's wearing that here. So let's get a little bit into the science and the statistics of the analysis. So this data is from the ENIGMA lifespan group. So they are interested in how do the brain measures that we can derive from imaging vary across the lifespan. And so there's, if you just look at the size of the brain, we know that increases till age 20 or so, and then it sort of declines gently till age 60, and then declines more precipitously after age 60. And you could just compute the size of the brain from MRI in data. Here's 18,000 people's data from all over the world. And you could ask some questions. You could say, well, do women and men differ in how they age? And there's some interest, maybe women, at least in terms of brain morphometry, decline more slowly than men until age 60, and then maybe catch up a little bit later. Or you could stratify this by disease or by genotype. And if you look at the data, this is really showing you how it's built. So the colors on there, each cohort, I mean, you know of imaging studies such as ABCD or ADNI or the UK Biobank. Each study is contributing to different parts of the lifespan. So if you look at the little colored dots, one center might be only scanning elderly people or maybe scanning adolescents. And essentially by piecing this all together statistically using regularized models, you can begin to get trajectories for different structures. So, excuse me, you can see for the thalamus or the amygdala or the hippocampus, the way in which they typically change with age is very different from one structure to another. So it begins to be really quite interesting. And you could imagine a sort of reference chart. So you could say, in women of a certain demographic background, what are the percentiles for volume of these different structures? And here you look at the data from women for the left hippocampus, and it's just the volume. And you can basically, in your own data, plot where you are. I mean, it may not be as important where you are in this curve as to what the trajectory is over time. And a person who's declining or not developing as you would expect, this would be interesting sort of reference data. So if you have the structural measures, you say, well, is there any way we can figure out how our genetic code influences this? So, I mean, there's a lot of things that affect development and aging. Some are not genetic. I mean, they are experiences and environment, but some of them are genetic. And so brain measures might be a good target for genetic analysis. It might be easier to find genetic variants that affect the brain. And this is an approach that's been pioneered by, for example, the Psychiatric Genomics Consortium, where they essentially collect DNA from patients and healthy controls. And they look at the DNA and look for markers that are statistically overrepresented in patients. So you could essentially do the same thing with brain scans. You would say, tell me where it is in the genetic code that markers influence, temporal lobe volume, hippocampal volume, the activity of the frontal cortex, or the molecular content, the amount of amyloid there is. And this is sometimes called the endophenotype hypothesis, because you're looking at a particular marker of a molecular process that you can measure more easily than the disease. And the idea is that might be more tractable for genetic analysis. Now, there's a little bit of a problem. So a prominent geneticist, Nick Martin, who's in this picture here with me, he said when this began that if you want to study genes that affect the brain, you'll need brain scans from 50,000 people. And I mean, he's a very famous geneticist. He was the president of the Behavioral Genetics Association some years ago. And he said that just because images are much more expensive to collect than measures of height or weight, it doesn't change the power calculations. And so let's call that Nick's bet. And so Nick's bet is that it's going to be difficult to find genes that affect the brain unless you have an absolutely vast amount of data. I mean, it won't be easy to do with, for example, 50 or 100 scans. So basically when Enigma was launched, the first studies that were done were to answer this question. They were asking, are there polymorphisms or genetic variants in the general population where they are associated with, for example, the structure of the brain? It could be the size of the hippocampus or the thalamus. And this paper in Nature, it's a chart of an effort to discover polymorphisms or variants in the DNA that influence the size of different parts of the brain. And I'll take you a little bit through this paper to explain how it was done. So let's say you can measure something from the brain. So on the left there, it is the phenotype. It could be the size of the brain or the hippocampus, or like we said, it could be a measure of the amount of amyloid or tau in the brain from PET. It's just something that you've measured in a population. Now you could say, I think the genetic sequence of these people has something to do with the measure. And so if you look at the genetic code of different people, there are points in the DNA or single nucleotide polymorphism where at least a fraction of the population has a different spelling, a different nucleotide. And so you could go along the genetic code after a step called imputation, which sort of puts everyone's genetic data on the same reference panel. And you could say, tell me if any of these letters or nucleotides in the genetic code is statistically linked to the measure that we measured in the images. And so in the little plot on the right, it could be that if you had a C and a C in the genetic code, the average intracranial volume could be a bit lower than people that have the AC genotype, or for that matter, the AA genotype. And I mean, maybe this doesn't happen very often in the genome, but if there is a place where it does happen, we should be able to detect it. So this has actually been done long before brain imaging genetics to try and find hotspots in the genome that confer risk for degenerative diseases such as Alzheimer's and Parkinson's. And I just want to explain this diagram. So the diagram here is a Manhattan plot. And along the x-axis are the common variants in the genome from chromosome 1, 2, 3, 4, all the way up to 22. And the little skyscraper things there, that's why it's called a Manhattan plot, because it looks like the New York skyline. If they're really tall, it means that variation at that locus on the genome is extraordinarily linked with disease. In other words, people that have the illness, in this case, Alzheimer's disease, are much, much more likely to have a variant at that point in the genetic code. And the y-axis just tells you how strong the association is. So the y-axis is actually the negative base 10 logarithm of the probability that this just has occurred by accident. And I mean, obviously, something like the ApoE variant, it's something that was being discovered long before genome-wide association studies by a linkage analysis. That's a genetic variant that triples your risk for Alzheimer's disease for every copy that you have. And if you look at it here, I mean, the probability is 10 to the minus 40. I mean, extremely unlikely that this is an accident. And then if you look at this, there's a list here of maybe 20. There are now about 40 Alzheimer risk genes. And this is interesting because you can then ask, what does the gene do? Does it influence the buildup of amyloid or inflammation or cardiovascular health, or any number of things that might contribute to the disease and that we might be able to interfere with or prevent? Now, you could say, well, I don't know that matters very much. I mean, my risk for disease might depend on what I do and a number of other things. But in fact, it does matter a lot. So this is work by Rahul Desikan, where he said that if you took all of those markers in the Alzheimer's disease genome and you sort of made a score, he calls it a polygenic hazard score, and you weight them by how dangerous they are. If you fall in the upper 1%, or for that matter 20% of these scores, your risk of getting Alzheimer's disease at the age of 65 or 70 or 75 is vastly higher than the average. And I mean, you can see as you get older, it's almost inevitable for people with some risk profiles to get Alzheimer's disease. But it's very interesting. There's people with, for example, the ApoE2 risk haplotype, which is found in about 10% of us. And they rarely get Alzheimer's disease at all, very much lower risk. And so it'd be interesting to know, I mean, how does this gene affect the brain? Is it preserving the hippocampus? Is it reducing the level of amyloid? Or is it something entirely different that we can't see on a brain scan? And you can do this in other diseases. So in schizophrenia, there's been genome-wide screens that implicate genes in the immune system, the major histocompatibility complex region in the DRD2 dopamine receptor subunit. And you can imagine that essentially as you get more data, actually this is an animation which I don't think will play, but these sort of towers, they're called climbing towers, they grow with more and more evidence as the amount of data increases that implicates certain hotspots in our genome in the disease. So ENIGMA essentially did the same thing. And so in ENIGMA, the members compute brain measures from their scans. It could be the thickness or volume of parts of the brain, maybe with FreeSurfer, or it may be the microstructural integrity or other measures from the white matter computed with, for example, FSL. And then each site that's taking part is asked to run a genome-wide association study. So precisely the study that I just showed you in Alzheimer's and schizophrenia, where they test if there's any evidence of statistical association between up to 1 million markers in the genome, these variants, and the things that they measured in the brain scan. And then to sort of combine everyone's results, we do a meta-analysis. So if you're contributing 100 scans, obviously the vote from your data might be a bit different than a site that's contributing 4,000 scans. And a meta-analysis just allows you to combine statistical evidence from multiple studies, I mean, in this case, they're coordinated studies, to reach a decision on the overall evidence that a marker is linked to some feature of the brain. So this works. Here's a map of the hippocampus. There are areas of the genome, particularly this harakiri gene on chromosome 12, where people with a certain spelling or certain genetic variant that's very common have a smaller hippocampal volume than the people who don't. And because we can do statistical parametric mapping or surface-based parametric mapping, you can actually run a statistical model in the image to tell you exactly where it is that gene appears to be erasing volume. So it's very, very interesting. And you can ask other people about what the gene does. So this is very interesting work by Professor Kazima Bulayeva. She's a Russian geneticist. And she studies ethnic isolate. She studies people that live in isolated mountain communities here in Dagestan. And she had found that these populations often have genetic variants that are linked with, for example, mental retardation or with psychosis. And she got in touch with us and said, the gene that you found where a variant appears to be associated with hippocampal volume, that's an area of the genome that we found is very, very different in people with mental retardation in this community we study in the ethnic isolates. And so, I mean, you can really imagine how extraordinary it is to have a communication like that from the other side of the world implicating the same point in the genome. So there's a nice story emerging of why that genetic mutation might link to mental retardation via a mechanism in the brain that you can map and understand. So these are very large-scale papers. This was one that discovered 6 loci in the genome that influence hippocampal volume. And you could ask about famous genes. So we talked about ApoE. A quarter of us carry at least one copy of ApoE4, which is the variant that increases our risk for Alzheimer's disease by 3 times. So you could say, well, tell me how the carriers of the E4 gene differ in hippocampal volume all over the world. And so there's people that are 20, people that are elderly. I mean, maybe the risk gene for Alzheimer's makes a huge difference late in life and has hardly any effect early in life. And that's exactly the case. And so if you look at the little picture in B, the graph shows the mean age of the cohorts or the studies that were in that ENIGMA initiative. And the Z-score on the y-axis is how much hippocampal volume is shifted by people that carry the ApoE4 Alzheimer's gene. And as you get older, there's a shift of about 2 standard deviations from the mean in people that carry ApoE4. So it's very interesting. You could study protective genes, you could study new genes that people have just discovered. And very often you'll hear in the news, new autism-related gene discovered. I mean, you could just look up in this data if there's any shifting of interesting brain measures and gather some insight into what might be mechanistically related to that diagnosis. So you could build genetic dictionaries of the brain. So for the hippocampus at the top, or for the intracranial volume at the bottom. Once you have maybe 30,000 people, as Nick Martin suggested in Nick's bet, you start to be able to reveal the genetic architecture for these different structures. And some of them are interesting. So one of the genes that has a variant that predicts brain volume, very famous gene. So the microtubule associated protein transporter gene, MAPT, which you see there, that's marked, that was already famous. So it's the second to highest risk conferring gene for Parkinson's disease, or at least it was in this genome-wide screen in 2014. And it's sort of intriguing that you can begin to connect the dots between genes whose function is known. I mean, in Parkinson's, synuclein alpha, which is involved in synaptic vesicle release of dopamine, there's variants in that gene that influence your risk for Parkinson's. But it also seems like there are genes that affect the physical structure of the brain, and maybe the trafficking of intracellular proteins. MAPT is known to do that. And there might be certain fractions of the population that have these variants that alter their brains and alter their disease risk. So all the subcortical structures have been mapped. It was a really remarkable effort to see if the genes that are associated with their volumes are the same or different. They're largely different. I mean, you can imagine that certain genes would matter for all the structures, but the evidence is somewhat the opposite. And you can travel around the surface of the cortex into different functional areas. And we're pointing in this example to the pre-central and post-central gyrus. And you'll see that the genetic variants that influence one are not exactly the same as the genetic variants that influence the other, although there is one that's in common. And you could really make a map of the whole genome in which parts of the brain they affect the structure or function of. You could make a sort of dictionary that refers to the known cellular functions of these genes, or how much they're implicated in different psychiatric disorders. And some of them, just to sort of tell you what they do, many of us from your childhood may know this is a slinky. So many of them, the genes might alter the rate of division or the pattern of migration of ventricular ependymal zone cells during fetal development. So if you can imagine, the genes that regulate where cells go in the brain might lead to differences in the surface area or thickness of different areas of the cortex. So there's a lot of work going on in mouse looking at homozygous genes to see if this is in fact the case. So this was all put on the internet. You can look at different genes and see how they affect the brain. So if you just discovered a gene, or you were interested in a gene, you can then go and look up how it's altering the brain based on this massive survey. Now you could say, well, Paul, that's all very good, but what about the function of the brain? So here's me in a hotel room in Moscow having an EEG, which is a very, very unusual thing to do. It looks at the synchrony of brain activity in different frequency bands. And Dirk Smit, who's a geneticist in Amsterdam, leads the EEG working group for ENIGMA. He and his colleagues found that in much the same way as the volumetric measures have genetic variants that associate with them, these functional measures do as well. So, I mean, one example in this very, very nice paper, it's the first study that we know of that combs the genome for markers that influence EEG, even the oscillatory brain activity that you can measure with EEG seems to be linked with variants in your DNA at certain locations. And if you stop for a minute and think of how remarkable it is that that can be found, I mean, it's just utterly remarkable that is possible to discover. And some of the genes make sense. So the GABA2 gene, which is in the GABA-A signaling neurotransmitter pathway. GABA, as you know, is the major inhibitory neurotransmitter in the brain. There's a polymorphism in this gene in that pathway that affects beta power in the brain. Beta is just a frequency band for oscillatory activity in the brain. And this was a known genetic marker that was associated with alcoholism and with epilepsy, but it wasn't clear why. And now at least we have some new leads on the mechanism. You could say, look, the activity of the brain is altered in this area, or some of these areas might overlap with known psychiatric disease risk loci. Now, another variant which is really interesting is to say, well, let's say I come back for a brain scan in a couple of years, and you measure the speed of my aging, or if you were a child, the speed of your brain's growth. You could ask if there's any markers in the genome that influence the speed of aging, the speed of development longitudinally. I mean, this would be really useful information. So this project by Rachel Brower and Hilke Holzhoff-Poll and the ENIGMA plasticity group looked at 10,000 people between ages 4 and 99 from 37 cohorts around the world, and just measured the rate of loss of hippocampal volume based on sequential scans, or the rate of expansion of lateral ventricle volume, which you can see is kind of bad news. If you're about 60, there's a massive speed up in the ventricular expansion rate and a massive drop off in the rate of hippocampal volume loss. But that's interesting. What causes it? I mean, maybe there's some markers that speed that up. And here's the result. So they map this based on longitudinal morphometric measures for different structures. The graphs are the rate of growth or loss for the different structures. And they made this wonderful dictionary, almost like a Rosetta Stone of the degenerative and developmental profiles, in which they implicated very, very specific genes that alter the speed of growth and aging for different people around the world. So very, very interesting data. Now, the mathematicians would say, well, why do you do this? So this is sort of genome-wide association. Isn't there some principle that we could understand? And if we could decode how the genome codes for connectivity or functional synchrony, I mean, it might not just be a question of mapping single nucleotides. It might be sets of genes or sets of markers in some sort of combination that interact to produce the brain structure. And so this analogy, and actually the name ENIGMA comes from the wartime effort to break the ENIGMA codes with a machine that you can see on the right. And some of the methods are actually rather similar to the ones used in code breaking. So here's one that uses distributed computation and deep learning. And it tries to find what combination of genetic markers in combination predict what combination of changes in the brain. Many methods to do this. Canonical covariates analysis is a classical linear method. There's other methods related to polygenic risk scores that add up contributions from multiple genes. Or you can use deep learning. I mean, you'd send a deep learning model to all of these sites and see if you can train it on the data, which is a very interesting project that we're doing at the moment with Vince Calhoun and Jess Turner, who lead the CoinStack distributed machine learning project. So I'll briefly tell you about some other kinds of genetic variation. So common variants are not the whole story. There are copy number variants, and the commonest disease that you might know about where this is important is Huntington's disease. So, excuse me, there are sort of triplet trinucleotide repeats in patients with Huntington's disease that influence their risk and age of onset for Huntington's. And if you look at these, even in a biobank of largely healthy people, there'll be 1 or 2 people, or often 10 or 20 people, that have rare deletions or duplications of lots of nucleotides. So we're not talking about single genetic markers, we're talking about chunks of the genome. And you can categorize these by chromosome, and some of them are more common than others, and you can rank them. And one of the ENIGMA groups called ENIGMA CNV is actually looking at these rare deletions. They have much, much larger effects on the brain. And so typically, I mean, there might be a genetic disorder such as Fragile X syndrome or Williams syndrome, or for that matter, 22q deletion syndrome. And the individuals with these disorders, they don't all have the same genetic deletion, but they do tend to have common patterns of effects. And so, I mean, this is a project you can see here that's mapping the selective vulnerability of different brain regions to different CNVs. Another type of epigenetic variation is methylation. And so ENIGMA's epigenetics working group studies how our genome is methylated. So there's areas called CPG islands, the cytosine guanosine regions tend to be methylated. And that is a reversible chemical modification of the genome that influences gene expression and therefore risk for disease. And they remarkably found, using the same genome-wide screens to look at the epigenome, that there were areas in the genome where the amount of methylation is associated with, in this example, hippocampal volume. So it's very interesting. So not just the genetic code, but some of the things that happen during our life that alter methylation of the genome is detectably linked to hippocampal volume. And they needed about 3,000 individuals' data, genome-wide methylation data, and brain MRI scans to find this. So that's very, very, very cool results by Sylvain de Rivière at King's College London and Tianye Jia, who was working with her to discover this. Okay, so last 15 minutes. So we talked about genetics and ENIGMA, and one of the ways ENIGMA got started was really born out of frustration, because none of the people working individually could find consistent evidence for genetic markers that affect the brain. But you could use the ENIGMA model or this sort of meta-analysis of images to reveal things about diseases, even diseases where we don't have any genetic information at all. So ENIGMA also pulled data from patients with schizophrenia, bipolar disorder, major depression, post-traumatic stress disorder. There's an ENIGMA addiction group that studies the effect of cocaine and cannabis and methamphetamine and opiates, and for that matter, cigarette smoking, nicotine on the brain, and alcohol. And then each of these groups clubbed together to make maps of the aggregate effects of these disorders, and then later on to compare them. So let's have a quick look. So this is a picture of the effect of each disorder on the cortical gray matter. So let's have a look at them one at a time. So major depression, there's not a great deal of difference. The subtle limbic system differences in major depression, the level of affect, which is influenced by the cingulate and other parts of the limbic system, affect is altered. In bipolar disorder, there are also, as well as depressive episodes, there are sort of manic episodes as well, which are thought to be influenced by the function of the prefrontal cortex, which you see here is also altered in patients on average with bipolar disorder. And then in schizophrenia, which is much more severe disorder in many cases, where there's psychosis and often disordered thought, many, many more areas of the brain seem to be altered, at least structurally, in patients. Not every patient is the same. So these are composite maps. I mean, a person with schizophrenia doesn't have to fit this pattern. They're just aggregate patterns. There are neurogenetic disorders. This one, people with this syndrome are at higher risk of schizophrenia. They have a very, very different pattern of excesses in gray matter in many brain regions. And then in the different forms of epilepsy, medial temporal lobe epilepsy, idiopathic or generalized epilepsy, the pattern of gray matter differences is very, very diffuse. And I mean, you can look at different disorders, and as we look through these, we can look at them all together. You can begin to see signatures. And so it's certainly the case that some disorders have quite selective correlates in the brain, where others are more general, and the overlap is very interesting. So, I mean, just one example, the cortical thinning associated with depression is fairly limited to the limbic system here, the cingulate, whereas in bipolar disorder, this map based on 5,000 people shows that there's a much more pervasive deficit in grey matter volume in the frontal cortex, which is involved, as you know, in inhibition. And you could make a sort of signature for each disorder. This is a study by Enigma of the disorders you see there, based on 20,000 people from 33 countries. And the little bars show you, for different structures of the brain, the lateral ventricles or the caudate and putamen, how much difference there is between patients with these disorders and matched, typically developing controls. And you'll see that schizophrenia is among the largest effects, bipolar somewhat half that effect, depression still smaller. And then disorders such as ADHD, or conditions such as ADHD, have a different pattern of influences that are really very distinct. So these results are all in an Enigma viewer built by Peter Kochanov and Katie Hatch and Zhan Chen. And you can think of sort of future directions for this. So we talked a lot about structural MRI. You can look at the genetics of fiber tracts. And so here's some fantastic diffusion imaging data collected at the University of Minnesota by Camille Ugerville and his team. I mean, look at the extraordinary detail. I mean, it'd be interesting to know how the disorders affect the fiber microstructure, how the genetics affect the migration or connectivity here. And so you could, as you know, with tractography, build maps of connections inferred from diffusion imaging. You could relate them to the cortical areas that we've talked about. And you could ask, are there any connections or pathways whose integrity is influenced by the genes that we have discovered? Or the other way around, you'd say, tell me, what is the genetic architecture of the neural pathways of the brain? You could build all of these things. And there are new studies in this area. So Enigma, just this last week, had a call about a very, very large-scale genetic study of diffusion imaging, where you could look at particular pathways. You could imagine a sort of connectome-wide, genome-wide screen. I mean, there's enough data now to do this, where you break up the brain into all of these different pathways and units and ask questions about the common and distinct genetic variations that are going on. So last 5 minutes, I wanted to say something a little more cultural and philosophical. So obviously, from working with people that are all over the world, I mean, there is a notion of collective intelligence. I mean, we can't understand any of these things fully on our own. But also, I think it's very important for science. I mean, Enigma has activities in 45 countries now. It's politically beneficial when projects are led from different countries. We talked about the added statistical power and the efficiency and the computing, but it's not just the resources that are being brought to bear in terms of capital. It's just the sort of range of backgrounds and diversity of people with different experiences that might have good ideas. And so that's been very, very important. We have activities of Enigma across the Russian Federation in Novosibirsk, in the academic city. Projects set up by one of our colleagues at USC, Vladimir Zelman. We have projects on HIV that many people in Thailand, for example, are contributing to. There's an India Enigma initiative to look at protective factors for brain aging that may be different in India compared to Western countries. Very, very interesting initiative. There's work in Japan, very interesting study led by a group called Kokoro, a consortium in Japan, that's found that the brain correlates of psychiatric illness on MRI and DTI are largely the same in Japan as they are outside Japan, which is probably no great surprise, but it's very encouraging. And then finally, other parts of the world that we might not traditionally have political relationships, very, very valuable work going on here in the Middle East, in the Arabic world, in Qatar. There's a new Enigma initiative. And you say, why would this be interesting? Well, culturally, in many of the parts of the world I mentioned, drug and alcohol use is very, very different. In the Muslim countries, it's very uncommon to drink alcohol or abuse drugs. And many other things are different. So not only the genetic, but also the sort of environmental risks for illness are entirely different, or may not overlap very much from one country to another. And so their observations obviously give us some perspective to triangulate and separate factors that might be either protective or detrimental to health. So what are we doing at the moment? ENIGMA is doing harmonized analysis of diffusion tensor imaging all over the world now with multi-shell diffusion MRI. There's a lot of piloting of resting state functional imaging, which is a little bit more difficult to harmonize in an international setting, with initial work in PTSD and MDD. And then also, for those of you interested in deep learning, obviously ENIGMA is a very rich, diverse framework to understand whether there are clusters or subtypes of diseases, or predict treatment response from a lot of aggregated biodata. So there's a lot of mathematics involving distributed or federated deep learning that operate even on whole genomes now. There's a new project being planned this week doing artificial intelligence and deep learning on whole genome sequences to try and find the causal loci for some of these disorders. And again, even in a field where genomics has been done for many, many years in psychiatry and neurology, we're beginning to understand how the genes that are being discovered actually affect the brain. How do they drive its development and aging? And are any of these relationships different in Japan or India? Or there's a new project that's been submitted for work in Pakistan or in China, very, very active project with people in China to look at resting state data in ENIGMA, to really understand the social as well as genetic effects on aging and mental health. So do visit ENIGMA's YouTube channel. There's all sorts of interviews and tutorials with eminent geneticists and neuroscientists. Do join in. I mean, if you have an idea, I mean, certainly lots of areas that might be interesting to work on. And if you want to take part, there's a website. This is on the INI website at USC, where you can look at how you might want to join in. Even just to hear about the news of what's going on, you're very welcome to join, or even to get involved with analyzing or collecting data. So I think that is it. So a huge thank you. Obviously, thank you to you for listening, and to Meredith for the invitation today. There's also an inordinate debt of thanks to all of the scientists who for 10 years now have put all their hard-earned work in neuroimaging and genetics into this project. And as we said before, often groups of people that are not traditionally allies, at least politically, are working together for the common good, which I think is one of the main reasons we all do science, and is a great tribute to all of the people that are here that this project was possible. So thank you very much.